Hi, Jess. How are you? I was hoping you could introduce yourself and provide an overview of your work.
I'm doing well. My name is Jess. I'm a fourth year PhD candidate out of Queen's University in Canada. I've had the really unique opportunity to dabble in a little bit of everything from forensics to clinical work, and more recently, some work at the Smithsonian Museum of Conservation. But basically, boiled down, my thesis is tentatively titled Application Driven Advances Using Ambient Ionization Mass Spectrometry. I've been really fortunate in the sense that I've been able to dabble in three different ambient ionization techniques. I find that people who get introduced to this field during their graduate school career kind of have experience in one mostly, but my lab uses something called the liquid microjunction surface sampling probe, and it's a liquid extraction based method. And with my thesis at Queen's, I've been hoping to introduce a new step in the natural product discovery pipeline. Basically, what I'm hoping to do is introduce a step where we can prioritize and screen for potentially novel secondary metabolites or metabolites that aren't really that essential for health. And to do this, I've been stressing the fungal or fungi that I've been working with. I've also worked with some bacteria to see how, if I were to change their environmental conditions with the help of my counterpart who does all of the growing for me, to see how those profiles might change and just screen for all the new metabolites that may pop up.
Very good. So, full disclosure, I am not too familiar with the type of mass spec that you're using. So, I was curious why this type of analysis is a good fit for your research and why is it better in this case than other conventional MS approaches?
I think this is a really good question. And I think the answer is that it's not. I think that it's very situationally based. So I can't really explain it without delving into the background of natural product discovery, but I don't know how familiar you are with it. But it takes a really long time. And I had no idea before putting my feet in the field. So typically, from what I understand, there's kind of two approaches. There's one, which is what my work is more geared towards, where people will grow their microorganism in bulk. And that can take anywhere between a day, if it's like something like E. coli, a week. And so when I say in bulk, that's like a 4 liter Erlenmeyer flask filled. Once that is at a mature stage of growth, that's put to a rotary evaporator and rotovap and purified, and then brought to a UPLC for fractionation. And then you're fractioning a lot because it's 4 liters after the purification. You have to further purify and further fractionate to get down to maybe like a couple compounds that you aren't familiar with, because if you're working with these microorganisms for a long time, you get a sense of what's around metabolite wise. And then you have a list of massive to charges of things that you don't recognize. And then you then go to NMR. And so this whole process can take about a year. Some people it takes 2 years. And then at the end, you find out that it's already been discovered. And so you've really spent all this time, all these resources, solvent, um, I guess personnel costs as well, and instrument costs, just to find out that it's been identified already. And so what I'm doing is I'm hoping to introduce a new step in the pipeline where identify the metabolites that are present before we get to that chromatography step so that we can pre-prioritize the signals that we already care about to lower the chance of identifying what has already been discovered. To answer your question, I don't think it is better, but I think it's a really good, almost supporting technique to help mitigate the time and cost before we get to something like chromatography, because like chromatography would be to like be the big guns. You don't want to rush into it before you have an idea about what's going on.
That is a great explanation. I appreciate that. You touched on it when you noted your use of rotary evaporation, but do you have any other notes on your sample preparation? What's the biggest challenge or the most frustrating area of that process?
Yeah, for me, obviously, we would have to do a lot of almost proof of concept. So we do both of these, conventional and our approach side by side, to compare the two. In terms of our setup, ambient ionization is really nice in the sense that there isn't much sample prep, but I will say that I experience a lot of clogging in our probe that we use. But Other than that, I guess on the more microbiology side, it's really the extraction step and the chromatography step that takes up the most of our time. Something that I read recently actually is that the extraction step is 80% of the processing time in natural product discovery and 99% of the solvent usage. So being able to have an idea before going into that step is really useful.
What my company specializes in is nitrogen blowdown evaporation, concentrating samples anywhere from 50 mil, the starting volume, and then getting it to a 2 mil GC vial or to dryness. But yeah, one of the hot topics in extraction for as long
Mhm.
as I've been in the industry is how do we do this with less solvent? So
Absolutely, yeah.
I am not surprised to hear that at all. Would you be able to talk about the challenges in identifying and confirming the structures of the metabolites?
Yeah, admittedly, I'm only able to say that the structures that we've identified are quite putative. We haven't really separated and done the NMR of them, but we're not as long as that there's a hit, we're not interested in it. Because if we're looking to find something more novel, then what's the point if we have a really high likelihood of even remotely having a match? But the trickiest part has been, frankly, tricking a lot of the LC based and chromatography based algorithms into thinking that I am doing a LC run. Because obviously, my chromatograms are not actually chromatograms, and the data that I have aren't actually what it think it is. And so a lot of my thesis is spent tricking algorithms into thinking that I'm doing an LC run when it's not. So that's been quite interesting, and I've found pretty unique ways of approaching that.
How do you envision research in this area contributing to streamlining, for lack of a better term, natural product discovery in the pharmaceutical research?
It's a really good question. I am really hoping that it really does streamline the prioritization. I think if instead of researchers going in blind and just having a list of things that they're not too familiar with, um, I'm hoping that the solvent use, which we touched up upon, is really allocated to the metabolites that we really care about. Yeah, so I'm not sure how quickly something like this will get adopted in industry, but I will shamelessly plug that I did 24 well plates with 13 different conditions in 6 hours with 150 mils of solvent. That takes me about 4 minutes a plate. And I know that I do it on 24 well plates, so although they're not in bulk growth, but it's enough information for us to see what it will produce in that little, I guess, altered environment. I'm hoping that something like this will allow researchers to not only introduce more conditions at once for testing instead of just focusing on the one, but also, again, like save solvent costs and personnel time. Our lab is also working on automating it. And so if it gets automated, then that'll be even faster because I've been doing it by hand. I really think it'll help prioritize like potentially novel compounds.
Based on your findings about light conditions affecting metabolite production, how could future work explore other environmental factors that might influence fungal secondary metabolism?
Fungi are really interesting. I really think that they have a behavior of their own. And I think that when I say fungi, a lot of people think mushrooms. I'm talking about mold. Something that I haven't presented on at the ASMS is that we're also growing them in a co-culture environment. So we're putting them in pit fights against other organisms to see what they produce when they are cornered. So I definitely think that is a different condition that could be used to kind of study other metabolite production. I've dabbled in altering the carbon growth conditions. Um, Fungi are really known for kind of carbon cycling in the environment. So there's definitely something there with like biofuel production. I've done some nitrogen conditions about adding some ammonium into it, like ammonium based. I think they come from fertilizers. We've also done pH, different temperatures. But something I'm also really interested in is phosphate based additives, even some transition metals being added into the growth media. I'm not sure how they'll react. I can't remember the exact statistic, but I think it's something like only like less than 10% of all metabolites in natural products have been discovered. And so there's a really big field here where really endless on what we can do to stress them out and hopefully produce new interesting metabolites that might be relevant to clinical work and other really interesting fields.
You mentioned ASMS, which is where you and I met this past summer. And that's really the hope of this interview series, is because I meet so many interesting researchers like yourself who are doing work, whether it's chromatography, mass spec, and the involved sample preparation. One thing I was hoping you could touch on that you mentioned before I hit start recording was the debate regarding validation through chromatography. If you could talk about that.
Yeah, absolutely. My personal opinion is that's very situationally based. I definitely think that in terms of quantitation, I don't know if, at least for the work that I do, if it's there yet. If anything, I think the liquid microjunction surface sampling probe, my lab likes to say that it's semi-quantitative. There are a lot of discussions about matrix suppression, even dilution, because it's a liquid-based extraction approach. So I don't think the technology is there for quantitative purposes for at least what we're doing. So I definitely think chromatography-based approaches to validate would be really useful. But I think for something like a screening application like what I'm working on, I think it's nice to have chromatography to validate the work. But if I'm just screening, like my priorities are elsewhere, not really the separation. I'm really just looking to see what's inside the samples. I'm doing it because I think it's really useful information to have. But if I find it, then isn't that enough to not have to focus on it or like the metabolites since I just want to find what's there. And so if there's even remotely a hit in a database or something, I'm putting it to the side and not really caring about it anymore.
I
Yeah, it's kind of
understand.
interesting, but I don't know. I think there's a lot of different opinions, but I'm not sure what everyone else thinks.
Was there anything else from your research poster that you'd like to discuss?
Not that I can think of, but the questions were really good.
Awesome. Would you mind discussing what you're telling me about your museum experience? I thought that would be pretty interesting to talk
Oh,
about.
yeah, sure. I was a pre-doctoral fellow at the Museum of Conservation at the Smithsonian in DC. It was my first time living in the US, and it was also my first time in a setting where it wasn't quite industry, but it also wasn't quite academia. It was government. And my project there was about finding formaldehyde traces in these fluid preserved specimens. When you think about jars and jars of like specimens that are fixed with formalin. And the Smithsonian is home to, I think, the world's largest collection of these specimens. But the thing is that a lot of the work being done on sequencing, it can't really happen because formalin does something called cross-linking the DNA. And so it basically, if you were to unwind DNA, it's like the ladder. But what it does is that it introduces almost new steps in the ladder and also puts them at different notches in the ladder, making the DNA sequencing quite difficult. And so they have these logs in the archives about how they were treated. And so what they wanted to know, is there formalin in these specimens? If there isn't, then the likelihood of being able to DNA sequence them for their genetic information is a lot higher. Same thing as what I do at Queen's. It's really expensive to DNA sequence and it takes a really long time. And so to spend all that money and personnel costs to find out that you can't get any genetic information at all is a pain. I developed a assay using a substrate-based ambient ionization approach called coated blade spray. It's from ResTech. When I got there, I was really excited. It was a derivatization-based approach. And what I found out was that a lot of the specimens were actually being stored in the same jars. And I had information on how they were treated, but obviously, when things are stored in the same jar for centuries, even if they're not treated the same, there's going to be some signal. And essentially, I found formalin in everything. It's really hard actually to find a glass jar that fits specimens. And so it makes sense that all of them were stored in the same environment, but they've basically been in this broth for the last 2 centuries. And it was not surprising for me to find formalin in the ones that were listed treated with ethanol. It
No,
was
that's an
really,
amazing project.
yeah, it was really interesting. I got to meet and work with a lot of amazing people. I definitely recommend anyone who's in grad school to look at the Smithsonian's projects because they have a lot of internships available. And I was able to find a really great supervisor that also worked with ambient ionization, who I also met at the ASMS. And so they do a lot of like microscopy based methods, a little bit of isotope mass spec while I was there. The Smithsonian has a lot of research projects that I had no idea were available to me.
That's great to hear. I appreciate you getting into that. And thanks so much, Jess, for the time today.
Yeah, no problem.